This is the basic molecular structure of an ATP molecule, adenosine triphosphate. ATP is called the energy currency of the cell, and it's analogous to a battery in that energy is stored within its chemical bonds and can be released for use in energy-requiring reactions by breaking those bonds. The molecule is made up of an adenine, that wee chemical we hear so much about when we're talking about the bases of nucleic acids, and is attached to a ribose sugar molecule, as it would be in RNA. 3 phosphate residues are attached in a series to the 5' carbon molecule of the ribose, and they're called the alpha, the beta, and the gamma phosphate groups. The phosphates are attached to each other by anhydrous bonds. These bonds are the ones which store the energy and which release it when they're hydrolyzed. When the bond between the gamma and the beta phosphates are broken, 7.3 kilocalories of energy are released per mole of ATP. So, how do we get from the food we eat to the energy storage in ATP? And that starts off with glycolysis, one of those pesky biochemical pathways that most of us have done our best to forget, if only to let us sleep at night and not wake up screaming. The first step of the glycolysis pathway uses one molecule of ATP, since it requires energy, and the same goes for the third step. So at this point, we have 2 less molecules of ATP than when we started. Seems we're not doing too well. However, we end up with 2 molecules of glyceraldehyde 3-phosphate, and both of them continue down the pathway to pyruvate. Each molecule of glyceraldehyde 3-phosphate generates 2 molecules of ATP, so we've gained 4 molecules at this part. That gives us overall a net gain of 2 molecules of ATP in converting glucose to pyruvate. We've also stuck a proton onto 2 molecules of nicotinamide adenine dinucleotide, known as NAD to his friends, giving us 2 molecules of NADH. The pyruvate forms a complex with coenzyme A, forming acetyl coenzyme A, in the process generating another molecule of NADH, and removing a single carbon which becomes carbon dioxide. The acetyl coenzyme A then enters the tricarboxylic acid cycle, also known as the TCA or the Krebs cycle. By way of a number of enzyme-assisted reactions, we get 3 molecules of NADH, 1 of FADH2, which is flavine adenine dinucleotide, and 1 ATP molecule via GTP. This diagram shows where the metabolism of amino acids also fits in to the TCA cycle, and why amino acids play a role in energy metabolism as well as protein synthesis. Now, the NADH and FADH2 are redox cofactors in that they participate in redox reactions, donating electrons to the electron transport chain. Now, there are 2 important things to bear in mind when it comes to the electron transport chain. One is that it requires a membrane, and the other is that it converts oxygen to water. Oxygen is a very reactive molecule, producing free radicals which cause cellular damage. By attaching 2 hydrogen ions, i.e. 2 protons, to half of an oxygen molecule, water is produced, and this process requires 2 electrons to be supplied, and they come from splitting the hydrogen into protons and electrons. Now, in order to transfer electrons from NADH and FADH, the hydrogen has to be split into these protons and electrons. We know where the electrons go. Where do the protons go? The protons get pumped across the membrane, while the electrons donated from the creation of the protons are passed along in the electron transport chain. Now, this is where the importance of the membrane comes in. When protons are pumped across a double membrane, it results in a higher concentration of protons in the space between the 2 membranes, and this gives a concentration gradient which can also be described as a charge gradient, and it can also be described as a pH gradient, because remember, it's protons that we've pumped across. So now we get to the ATP synthase. This is a diagrammatic representation of the molecular structure of the ATP synthase enzyme. Notice that the dark and light blue parts, the F0 structure, are embedded in the inner membrane, while the red part, the F1 structure, is inside the cell, or inside the mitochondrion. Now what happens next is due to the structure of the membrane itself, which is a phospholipid bilayer. Now that is comprised of a gaggle of phospholipids which have hydrophilic heads and hydrophobic tails. What does this mean? Well, hydrophobic molecules are non-polar molecules. They are immiscible in water. Water is a polar solvent. The inside of our cells is water-based. Think of trying to dissolve oil in water. It doesn't happen. In fact, the interaction between the non-polar oil and the polar solvent forces the oil to form into spherical blobs to minimise the amount of surface area in contact with the water. When you put phospholipids in an aqueous environment, the hydrophobic tails attract each other because of the repulsive force of the polar solvent. And thus you end up with a bilayer where all the hydrophilic heads are on the outside and all the hydrophobic tails are on the inside. Why is this important? Now let's look at this part of the enzyme from above. The part of the ATP synthase embedded in the membrane has an aspartate at amino acid position 61. Aspartate groups are negatively charged and they do not like being in contact with an uncharged environment. An arginine group at position 210 captures one of the protons from this high proton concentration and hands it to the negatively charged aspartate. And this means that the aspartate now has no overall charge and it's much happier within the membrane. And the proton is then handed down the length of these protein subunits until it gets to the inside of the cell, and they are still trying to elucidate the actual steps in the process. Now the movement of protons is what causes the protein complex within the membrane to rotate. Think of what happens when you bring the north ends of two magnets together, one of them on a piece of string. The magnets repel each other, and so your dangling magnet will move away from the one you bring close to it. However, if you bring the south pole of the magnet in your hand to the north pole of the dangling one, the magnets will attract and they will come together. And that's a really good analogy to illustrate how the balance between the repulsive force of the negative aspartate and the lack of repulsion when the aspartate has captured a proton and can transfer into rotation of the protein complex. Now the rotation of this F0 part is not transferred to the F1 part of the molecule. The F1 part is held static by a stator protein. So the stalk of the F0 rotates inside the static F1 part. And that is seen as the light blue and the dark blue circle in the centre of the molecule. Here we are in effect looking at the F1 part from below. The rotation of the F0 inside the F1, as represented in these two wee blue dots, causes conformational changes within the F1 part. ADP and inorganic phosphate loosely attach in one conformation. When the asymmetric F0 rotates, it forces a conformation change in the F1, causing the ADP and the inorganic phosphate to be held closely together to allow a bond to form and ATP to be produced. And when the F0 further rotates, it causes yet another conformation change, which releases the newly formed ATP molecule into the cytosol of the cell. A good analogy for this mechanism is a water wheel. The flow of water downhill due to gravity, is able to turn the wheel. However, the analogy breaks down when you consider that the movement of the wheel is in the same plane as the movement of the water, which is the driving force. In ATP synthase, the movement of the driving force is at 90 degrees to the rotation of the F0 structure. Additionally, the rotational movement isn't being transferred to the F1 unit. In fact, the function absolutely depends on the F1 unit remaining static, so that the rotation of the asymmetric F0 stalk can force the conformation changes in the F1 subunits required for ATP synthesis. So that's how we convert the energy from our diet into energy which can be utilised in further metabolic processes. In other words, this is how we charge the little ATP batteries of our cells. As an aside, you'll remember the description of the ATP molecule being adenine attached to ribose sugar. Now here's a diagram of polymerized ribonucleosides, i.e. RNA. Instead of 3 phosphate groups being attached, as in ATP, there is one phosphate and then it's attached to another ribonucleoside. When we consider the idea that RNA may have been the first self-replicator, it's also easy to see how this energy currency could have come into being without the need for proteins. We know that ribonucleosides, left to their own devices, will polymerize into chains in the presence of phosphate, and that forms RNA. And we also know that the nitrogenous base and a ribose sugar will spontaneously bind to form a nucleoside. Now, I hope you've managed to follow on everything I've said. If you have any questions, please don't hesitate to ask your question in the comments section.